Thank you very much, Stephen, and chairs, and for Mort and the organizers to invite me to lecture today. And I was just going to say I appreciate the topic. I actually went back and had to review a lot of gene expression profiling data, but it actually makes you learn it even better. So with the beginning of the agenda, I'm going to just brief overview. The audience knows about large cell lymphoma, an overview about gene expression profiling. In particular, the two major techniques, Lou Stout's group was cell of origin studies, looking at unsupervised gene expression profiling to look at diffuse large B-cell lymphoma and trying to characterize them by looking at normal counterparts, and we'll explain that in a second. And they used primarily the lymphochip, whereas Margaret Shipp from Dana-Farber looked at supervised learning prediction method and looked at two different extremes. Patients were looking at groups that they were cured, about a 70% 5-year overall survival, compared to a group that had fatal disease where there was only a 12% overall survival at 5 years. And they looked at also thousands of genes, and they used the Affymatrix chip. Actually, the original Affymatrix chip that they use is no longer utilized, although they have other upgraded chips. The primary findings from those techniques, though, gave prognostic stratification and predominant and able to identify novel targets and pathways, which we are now utilizing for development of new treatments. So based on that development of novel therapeutic agents, I'll mention them in more detail and some conclusions. We all know that diffuse large B-cell lymphoma is the most common lymphoma, peak incidence in the 6th decade. Large cells, loss of follicular architecture, can present as extranodal disease, curable about 50% of patients, maybe slightly higher with the utilization of rituximab. Median survival, if not treated, is mean. It is basically patients who are not cured of this disease. It is a fatal illness. Diagnosis, we know, is a B-cell phenotype. And prognosis, as we're going to discuss, one of the most important findings in recent years was the International Prognostic Index, which still has a lot of input today. We should be utilizing it, but also genetics and also the use of gene expression profiling, where we're not sure how to utilize it in everyday therapy, but now it's being implemented in evaluation prospectively now. Just to remind people, the IPI, AP, LES, age, performance status, LDH, extranodal, involvement more than one extranodal site and stage, with respect to being able to determine with respect to 5 and 10-year survival between low intermediate, and high. Essentially, the IPI, though, is a representation or a surrogate for the actual biology of patients' individual lymphomas. And I think that's just important to note, is that it does not give us what is the mechanism responsible, but it does give us the ability, whether we are in large cancer centers or small clinical centers, in private practice, to be able to estimate what the patient's IPI is and perhaps actually guide the therapy, whereas it used to be one shoe fit all, that one therapy was utilized, everybody got chopped. We should be actually making decisions at the time of diagnosis and actually stratifying our patients to different therapies as needed. With respect to gene expression profiling, using DNA microarrays is a major technological advance that has increased our knowledge of the biology of lymphoma. It can identify and classify lymphoma subtypes. It defines prognostic predictors. It identifies potential novel therapeutic targets, and its knowledge is now being applied in the clinic. One of the first publications was by Elizadeh Shipp. This is actually Lou Stout's group. It looked at a complex but an ordered picture, a variation of gene expression profile or patterns and lymphoid neoplasms. This with respect to 96 different lymphoid neoplasms, including large cell. And they compared the expressed genes, and they looked at the normal lymphocyte cellular populations. And then with that, we can make a differentiation between cells that resembled more normal germinal center B cells, as well as looking at also activated peripheral blood B cells and other subtypes. It gave an insight to the biology of these neoplasms. And then work by Roswell, which refined this a little bit more further, took a look at and determined that there was at least 3 diseases with respect to large B-cell lymphoma. A germinal center B-cell type, which actually, a couple mechanisms, BCL2 translocation, CREL amplification. Actually, reviewing the literature, CREL amplification was noted as being, increased in germinal center, but actually CREL, it's not the actual CREL that's functional, which actually goes along with NF-kappa B upregulation, which is in the next one, activated B cell, is constitutive activation of NF-kappa B. and it's also a post-germinal B center. And then the third, the primary mediastinal B-cell lymphoma, which is believed to derive from a thymic B-cell and also has separate oncogenic mechanisms. The two groups, germinal center and primary mediastinal, being considered favorable, whereas the activated B-cell de novo at the time of original diagnosis is considered to have a worse prognosis. So molecular features of the GCB subtype, they express levels of genes which correlate to normal germinal center B cells, as mentioned here. The functional effect of several of those mechanisms is blockage of apoptosis. Therefore, agents that can affect either apoptosis, BCL6, or P53 modulators may have a better activity in GCB. With respect to ABC, their expression pattern of genes is more similar to activated B cells, NF-kappa B being the master transcription factor, which is constitutively activated. And I'll show you a slide in a minute on that. But I think it was interesting to make sure we understand that NF-kappa B actually can transactivate over 200 genes, which encodes factors for cell cycle, cytokines, growth factors, and apoptosis inhibitors. And its signaling complex can be mutated. Not only that, but also mutations can be seen in the BCR signaling complex affiliated or associated with this group of lymphomas. Potentially active agents will target the BCR signaling pathway, and there's an inferior prognosis not only with original data that looked at CHOP specimens that patients had received CHOP, but also in the R-CHOP arena now. This is just a very nice, it's a little bit active, but it's a slide by Lenz and Stout in 2010 looking at the B-cell receptor on top, as was mentioned also by Dr. Keating, with the normal B-cell receptor. But below is with respect to mutant CARD11, which actually demonstrates that you have a change with respect to the receptor actually internalizing and being constitutively put on, or you have a chronic activation of the B-cell receptor signaling. The bottom line is that the B-cell receptor may be one of the most exciting arenas of actual research and actually therapy that we can be giving patients in the next 5 to 10 years because of being able to target that, which actually drives primarily the activated B-cell type, but also other maybe subtypes of lymphoma and other malignancies. Moving around a little bit, with respect to the work that was done by Ship, I think it was very interesting that in her supervised analysis model, whereas the Stout model and the Rosenwald model looked at maybe over 100 some genes, but 17 were the most important one, they found that 13 gene outcome was the best predictor. The unfortunate thing of those 13 genes that were mentioned in this Nature Medicine paper, many of these did not have antibodies for the immunohistochemical staining for proteins representing these genes. The most important one, though, is protein kinase C beta, which is a predictor of unfavorable outcome. As mentioned before, this is a heat map, red being hot, the dark blue being cold. Those patients being cured on the left upper hand, those are genes that were overexpressed, and in the right lower half are genes overexpressed in those patients that had fatal refractory. One of those genes listed there is the one that they mention most, which is the protein kinase C beta. Now, what's important, and we should not ever forget, is that it's not the tumor cell alone. Often I do translational research. We isolate the tumor cells and study them, but the tumor cell does not survive in a vacuum. It also has influences with respect to its microenvironment. So this stromal gene signatures in large B cells, published by Lenz in 2008, they looked at over 400 pretreatment tumor specimens from large cell B-cell lymphoma patients, including those who received CHOP or R-CHOP. They did a multivariate gene expression-based survival predictor model, and the microenvironment was found, not surprising, to influence outcome. There was a stromal 1 signature which was favorable to show extracellular matrix and macrophage infiltration, and the stromal 2 signature, less favorable, showed that angiogenesis actually had a negative predictor value. Now, with respect to these there was additional work by Ship that showed there were consensus clusters. Using pretreatment large cell lymphoma specimens, there were 3 distinct subsets of large cell lymphoma identified. 1, oxidative phosphorylation, basically expression of genes that regulated mitochondrial function, apoptosis, proteasome activity, BCL2 translocations, and fast death domain deletions. So it allowed apoptosis protection, anti-apoptotic activity. There was a second main group of consistent clusters called B-cell receptor proliferation group, expression of components of the B-cell receptor signaling cascade and BCL6, And then the last, it actually correlates with they had host response group dominated by non-tumor cells in tumor microenvironment. In a minute, we'll discuss where therapies that are available today can be implemented based on this work on gene expression profiling with respect to maybe using, for example, the first one with BCL2 overregulation, BCL2 inhibitors. Number two, the B-cell receptor. The number of agents we heard in CLL are also applicable and have to be tested with respect to large cell lymphoma, and the host response with respect to the use of immunomodulatory drugs. The interesting thing is these three subgroups did not differ in patient outcomes, but did allow us to identify new therapeutic targets. This was a very nice, actually, slide and a review by Jonathan Friedberg from the University of Rochester. It actually looked at the B-cell receptor pathway members and looked at where the various agents actually can block the B-cell receptor in different points. We know that there was work has been done on SICK, that is basically the B-cell receptor. Downstream from the B-cell receptor is a number of targets as mentioned here, which include actually the BTK inhibitor, PCI, 32765, Cal-101 with the PI3K delta isoform, the AKT, or also the PKC with enzastorin, which we'll be talking about in a second. If we don't block those various factors in the B-cell receptor, what ends up happening, it just leads to increased proliferation, angiogenesis, and survival of the B-cell, in this case, the neoplastic B-cell. So I put development of these five major anti-tumor agents in large cell lymphoma was really based on gene expression profiling analysis. And we'll go through each one. NF-kappa B. With respect to NF-kappa B, you have several inhibitors of not only NF-kappa B, but also I-kappa B alpha. There's also people looking at inhibitors of NET8 inhibitors and other with respect to this area. There was data from a trial that I participated in looking at the combination of bortezomib and EPOCH in relapse refractory large cell lymphoma. It was interesting that the ABC phenotype and it was actually using the Hans criteria by immunohistochemical staining, which demonstrated concordance of around 90% with actual gene expression profiling. The samples that had both, you can analyze by gene expression profiling and IHC, immunohistochemical staining, showed an 83% response rate with the ABC, but only 13% with the overall response rate with the GCB, and the difference in overall survival. Believe that the ability to target NF-kappa B could actually reverse chemoresistance and resensitize the cells. And I think that's also important, that single agent activity with a number of these agents is not very high, but that it's what they do in synergy with other agents potentially can resensitize a cell to chemotherapy or resensitize it to killing. Lenalidomide in large cell lymphoma I've been working on for a number of years and studying it, and it appears that the ABC, or the non-GCB, actually has greater activity than GCB, we'll mention in a minute. And based on this work, there's a phase 2 R-CHOP plus bortezomib trial that was done. But what's interesting is that normally you would expect the non-GCB or the ABC to have worse outcomes. The outcomes basically was similar between the GCB and non-GCB, therefore suggesting that the non-GCB patients actually had a better outcome than expected. Based on this, there's a phase 2 randomized trial looking at R-CHOP plus or minus bortezomib in the non-germinal center patients underway. The second thing is the B-cell receptor signaling pathway. And with respect to that, one of the things is activating spleen tyrosine kinase SYK, which leads to downstream increase in BTK, PI3K, and AKT. So fostamatinib, it's hard to say, was found, this is SYK inhibitor, found to be active in B-cell receptor-dependent large cell lymphoma. The ABC phenotype had about a 30% overall response rate, and it looks promising in development in large cell lymphoma, not only by itself, but potentially in combination with other agents. In respect to other agents in this group, the BTK inhibitor, but also PI3K, in particular Cal-101, which is the delta isoform. By itself, these agents have had limited activity. For example, Cal-101 in 8 patients in a combination trial had 0 activity out of those 8 patients in large cell, but it wasn't studied what the patient's subtype, the genotype was, or the phenotype. So there are other trials that are in development I think will be important. A lot of the trials have not yet tested the cell of origin, meaning GCB or non-GCB. I think these things have to be done, and they're planned in a number of these trials prospectively to be able to analyze if different groups actually have better activity. Going next to the PKC beta gene, it's overexpressed, as I mentioned before, in the SHIP model with the fatal refractory large cell lymphoma group. It's associated with poor outcomes when you find expression of the protein on the surface of the cells, and it's associated with a decreased overall survival. It's downstream from the B cell receptor and NF-kappa B. It increases tumor angiogenesis via VEGF signaling. And one drug in particular that's a direct inhibitor is enzastaurin. It's a selective inhibitor, it has activity in vitro. In vivo, in a subset of patients, actually was tested, and there was 4 patients out of a group of patients that were tested and actually had 3 CRs, and one patient had stable disease for several months, multiple months. So it was a very small subset of real information, but basically right now there's a phase 3 trial. Reportedly, the phase 2 randomized trial was done, had showed some promising result, but a randomized trial looking at enzastaurin post-R-CHOP, either enzastaurin maintenance or no maintenance, to see if it works. That data is still pending. The next thing, angiogenesis, is very interesting to show that not everything that looks promising or that we have a target against necessarily is going to be able to be targeted by that agent or is safe. The angiogenesis arises from the stromal 2 signature, as well as there's actually data showing that high pretreatment VEGF serum levels portends that there has poor outcome in lymphoma, large cell lymphoma. So there was a question, maybe adding bevacizumab, Avastin, to CHOP or R-CHOP would be beneficial. However, it was not. And the toxicity concerns demonstrated that there was cardiac toxicity, left ventricular ejection fractions that were transiently decreased, patients having episodes of hypertension. There was GI perforations that occurred in patients and sudden death in 2 patients. With respect to patients receiving R-CHOP and bevacizumab and upfront therapy of patients with diffuse large B-cell lymphoma, there was also a trial looking at CHOP plus bevacizumab in T-cell, peripheral T-cell lymphomas that had to be stopped early as well due to toxicity concerns. But I think that shouldn't be that we should not study angiogenesis and anti-angiogenesis agents, because there's a whole slew of anti-angiogenesis agents with different mechanisms that actually could be studied in these patients. One of them being, for example, lenalidomide or the IMiDs, which actually have a pleiotropic number of activities, which one of them is angiogenesis. So next, the microenvironment. Well, IMiDs have anti-tumor activity, about a 30% response rate in those patients with large cell lymphoma from phase 2 trials. They're related to the multiple mechanisms of actions, and predominantly microenvironment, as well as anti-angiogenesis. And lenalidomide appears, as I mentioned before, to have better activity in the non-GCB or the activated B-cell phenotype more than the GCB. There's a global phase 3 trial ongoing now. And clinical trials are now also exploring whether the upfront addition of lenalidomide to R-CHOP as initial induction therapy in large cell lymphoma may be beneficial to improve outcomes, and or using it maybe in the maintenance either alone or in combination with Rituxan as a post-chemotherapy induction strategy. As just mentioned briefly, this has been, most of you have probably seen this before, it's to look at the effects of lenalidomide not only directly on tumor cells, but also in the microenvironment, on a number of cytokines being produced in the microenvironment, its anti-angiogenic effects that are clear, also its ability to increase T-cell, the T-cell immunity, as well as activate NK cells. This is just one algorithm. So what do we learn from gene expression profiling? Well, it's not really feasible right now that we'll be able to get a full gene expression profile in every patient that has large cell lymphoma. So if we can standardize and have a highly reproducible type of immunohistochemical assay in a limited number of, say, proteins such as this is the Hans algorithm, which can determine GCB versus non-GCB based on basically just looking at 3 proteins. I think that's actually very useful. The Hans criteria has been criticized in recent years, but in samples that have been well preserved and in using perhaps one central lab, there can be concordance rates as high as 80 to 90% compared to gene expression profiling. This is work that was done by our group at Roswell, that a retrospective study with respect to lenalidomide showed in the GCB phenotype, basically there was less than 10% responses. But in the non-GCB, we saw over 50% response rate with respect to the use of oral lenalidomide. And based on that, that is part of the phase 3 clinical trial. Right now, there's two phases. There's a stages. Stage 1, we're looking at patients. We're stratifying by the Hans criteria, GCB or non-GCB. And then the patients get randomized between lenalidomide or dealer's choice, a standard control. And then after the first 100 patients, we have about 80 patients done. The second series of patients will go on to go, non-go analysis, a randomized phase 3, which hopefully we may go back to the FDA to see if we can limit the number of patients having to go and get rid of the control arm. So this basically near the end here, these are the novel targeted agents for large cell derived from gene expression profiling. A number of these agents, just to mention, bortezomib, enzostorin, bevacizumab, lenalidomide, fostamatinib, the PCI, the CAL-101. We didn't mention in detail, but everolimus, which actually is an mTOR inhibitor downstream from PI3K and AKT. I also mentioned here the Genentech novel agent DC-S4968, which actually is a CD79B, which is the surface of the actually B-cell receptor. It's one of its components. It actually targets the surface of the B-cell receptor, and it uses basically the same agent that's being utilized in brentuximab vedotin. So with respect to using a tubular toxin, internalizes on the B-cell receptor, this actual, when it binds to CD79B and actually attacks the B-cell receptor pathway. So it's yet another way of actually targeting B-cell receptor. And there was a number of literature that was done by Ari Melnick in particular from Cornell, looking at small molecule inhibitors of BCL6, which I think may actually be promising. But BCL6 is not only in the GCB or ABC, it can be distributed between both groups, but still an interesting target for therapy. So in conclusion, large cell lymphoma still remains a fatal disease in a significant percentage of patients today. Large cell lymphoma has biological heterogeneity, not only by gene expression studies in our clinical trials and outcomes, but we also note that there is a role not only for attacking the tumor cell, but also for looking at treatments directed against the immune and stromal targets. Utilizing current therapy and data, we are able to identify subgroups of patients of large cell lymphoma that have optimal benefit from novel targeted agents. What we described in myeloma today, though, in addition to the cell of origin, we need to validate biomarkers, but every time we change our therapies, we may be changing the natural biology of the resistance or the cells becoming resistant. We need to revalidate actually whatever markers that we utilize to study these patients to determine if they're going to be more selective or active in one group versus another group of therapies. In the future, I predict that gene expression profiling and other more sophisticated, perhaps global genomic analyses will continue to improve our understanding of lymphoma and improve our outcomes for our patients in the long term in the future. Thank you for your attention.